Okay, good morning, students. I think I'm audible. I'm audible. Yes, shall we start the class? Yes, thank you, Aditi. Clarity is there, ma, Aditi? Clarity is there, Aditi? Shall I start? Yes? Okay. So now I'm going to do today, it's going to be a very interesting topic because we are going to do with what you call the sex determination, the mutation and the genetic disorders. It's going to be very interesting. So first we'll start on with the sex determination. So now we have to know the basic. Yeah, thank you, Aditi, for that feedback. So first basic, you should know that a number of cytological observations has been made. Okay, in a number of insects they have made it, and that has led to the development of the concept of various genetic and chromosomal basis of sex determination. So the sex determination was first they carried out in insects, and with basis of lot of cytological observation they came to know about what is the behind determining a sex. So the credit goes to Henking. Okay, the person is named Henking. So he was the one who were able to make he was able to trace some specific nuclear structure which was all through the spermatogenesis in few insects. So he observed that 50% of the sperm received that structure and 50% did not receive. So it was Henking who gave the name to that structure, that there is nothing in that structure like that when he made that X, that became the X body, but he could not explain the significance. So further experiments done by Henking, he observed, and it was later noted that in fact that is what the name given as X chromosome. So we have different types of sex determination. Okay, we have what is that actually to do with sex chromosome? So sex chromosome is nothing but it's actually the sex determination is a method by which we are going to distinguish between a male and a female that is established in a particular species. So it is all under the control of a specific set of chromosomes, what we call them as sex chromosomes, okay, or they are also called as allosomes. Okay, the rest of the chromosomes, what is present? See, we are having 23 pairs of chromosomes. So in that, when I say that, then the 22 pair, what is that? They are the autosomes. So the rest of the chromosomes are called autosomes, and the chromosomes, that is which is specific for that determining the sex, that is the sex chromosome. So there are basically, we speak with 5 main genetic mechanism of sex determination. Okay, now what are those 5 methods? Let me explain. We have what is XX XY method, okay, and then we have what is XX X0 method, okay, then we have ZW and Z0 method, okay, and then we have Z0 ZZ method. So very important exam question. Sometimes there is a case of haploid diploid method also. Okay, we will go one by one. So first, when we see with the XX XY method, so this XX and XY method, it is quite common with the Drosophila and humans. Okay, here when you look carefully here, the females are homogametic, that is they will have the same gamete, same paired gametes, they are similar, XX. What about in female? In case of males, they are heterogrammatic. Okay, so here, but the total number of chromosome in male and female will be the same, but here the females are homogametic, the males are heterochromatic. This point you need to know. Okay. So now this basic, like first you know, the other things I'll just explain. Then we have the XX-XO method. In this, okay, this is quite common with grasshoppers. Okay, it is quite common with the grasshoppers, certain insects and all. Here you see the XX. Here, this is a female which is homogametic. What about here in male? There are two types of sperms. It can be either X or not. That means X means what they are having, like they are having the, what is called, if they have X, we call them as gymnosperms. If they don't have X, we call them as androsperms. Okay. Okay, so they have two types, XX and X0. Okay, so this is another type what we know. Okay, so see here, if the eggs are fertilized by sperm that is having an X chromosome, it becomes a female, and those that is fertilized by sperms that do not have an X chromosome, that will become a male. Okay, so it is the involvement of the X chromosome in the determination of the sex. So that is why it is designated to be the X chromosome. Okay. Another thing, the rest of the chromosomes were named as autosomes. So grasshopper, as I said, it's an X0 type of sex determination where you see the males are having only one X chromosome. So they will be having only one, or that is they'll have X and not type. So that is, we say that what you call the gymnosperm or the androsperm besides the autosperm. But here the females will have two X chromosome. Okay. Same way, when you observe these things, like say in case of mammals including man, we have the XY type of determination where both men and women, they will have the same number of chromosomes. Okay, like how you see here. See, this is where we see. So here the X chromosomes, these are the Y chromosomes. Yes, as you all observe, this is quite common in humans and Drosophila, the XX and XY type. What about here, as you see in case of what you call in Drosophila also, it is the same, the XX and the XY type. But in case of birds, it's going to be quite different, where it is Z and ZW method. So you can see here in case of females, we have the heterochromatic here, and we have the homogametic in males, which is quite contrast to our way of sex determination. Okay. So you are able to follow. So far, so we have to understand here in one prospect, that is, if it is going to be a male in case of grasshopper, okay, how it should be, the male will be expressed as AA plus X0. If it is a female, it will be AA plus XX. So this is in case of the grasshopper. This is how the autosome plus the pair of the sex chromosome, that you need to know. Okay. Now here, as you see, the males as an X chromosome is present, but its counterpart is directly smaller, which we call the Y chromosome. So you can very well appreciate the Y chromosome that is quite smaller than the X chromosome. But you can see the females, they have two pairs. The females have two pairs of X chromosome, right? So here both male and female, they'll have same pair of number of autosomes. So males will have, say for example, like how do we express it in case of males? So we have a pair of autosome. Okay, so this is how a male offspring will be, and in case of female, it will be XX. Okay, so while the females have autosomes plus XX, so that is how they will be represented. So usually you should know that the females, okay, they will have two gametes like XX, and males will have two set of male sperms, like it can be either X or Y, depending on which it fertilizes. It is going to, so they all are haploid. So depending on what is getting fertilized, it will determine the sex of the child. And at the same time you should know, okay, and at the same time you should know that what is that, the zygote, whatever is formed, it is going to restore the diploid nature. Okay, so here it is all N, the diploid nature is going to be restored. So it's probably by the fertilization of the ovum with the sperm. And it is always, you should know that same way in case of humans and Drosophila, the males will have one X and Y chromosome, whereas the females, they have a pair of chromosomes beside the autosome. So this is going to be the representation. Now we have to know that there is this X0 and XY type, right, which I already said. So this X0 and XY type, you can see here, in both these things, the males are producing two different types of gamete. Either they will be with or without X chromosome. So I said there will be two type, X type of sperm or not, that is androsperm and gymnosperm. Some gamete with X chromosome and some with Y chromosome. So it can be like that. So such a type of sex determination, we call it as male heterogamety. So we have male heterogamety. That means what? The males only are having two different type of gametes. That is, they can be either X and Y type or they can be X and not type. So what we saw in Drosophila, humans, those grasshoppers, okay, they are all or what type? They show male heterogamety. So see here. So these two types of these two methods of sex determination, they are going to show, 1 minute, I use another color. So they are going to show the male heterogamety. Okay, you are able to understand. So you can see that they are showing this. Okay, so that is where that is explained. But whereas there is another thing, like in case of birds and all, like when you go for certain birds and all, like you can see in birds, like the birds, it will be like AA plus ZW, which will be a female, and if it is going to be a male, it will be homogamete. Okay, so here in case of certain birds, it is totally different. That is, in this case, the total number of chromosomes is same in both male and female, but two different type of gametes in terms of sex chromosome, but they are produced by the female. So they show female heterogamety. So this is quite different with what the previous type we saw. So they are designated at Z and W gametes. Okay, you have one Z and one W in case of female. And if it is going to be males, they will have a pair of Z chromosomes beside the autosomes, like how it is expressed. Okay, so this you need to know. So this is found in birds, certain insects, fishes, reptiles, etc. So it is showing the female heterogamety. So there is also another method which we can say is Z0 and ZZ type, which is quite common with some moth and butterflies also. So here also the female will be heterogametic, like it will be expressed as autosome plus Z0 or it will be AA plus ZZ. So the gametes in female will be either Z or not, that is not type, or it will be a pair of homozygous. It will be a pair of what you call a pair of similar gametes. Okay, Z chromosomes you will see in the males. So this is quite common where we see in terms with moth and butterflies. I'm sorry, it's not homozygous. We'll use the word similar gametes. Okay, so that is where you have to understand. So again, I'm recollecting ma. See here, we have these. So why should a sex to be determined? I already said you can make clearly that it is a male and a female in a particular group of organism of that particular species. So we have sex determination of either XX, XY method, in which I said the males here are heterogamete. But in case of these birds or moth or butterflies, you can see that it is the female are the heterogamete. The female shows the heterogamety. So that point you need to understand. So there is also a haploid diploid method which I said, which is quite common, like what we study in case of parthenogenesis. So same way the unfertilized egg will develop into a male, what we call it as harkenotoky. Here what will happen, those unfertilized egg will develop into the, so here you can see that you have the males, we will have the females. Okay, so here the unfertilized males that will develop into the what you call the so here by the process of mitosis the sperms will be produced. In case of females, it will undergo meiosis and it will form the eggs. The unfertilized eggs, okay, that will develop into a male. That is in case we can give the honeybee example, haploid diploid method. So what is happening there, Aditi? There we can see that in honeybees, all the unfertilized male eggs, they become the what you call those, what are they called? We have those male bee, right? We have the queen bee, we have the worker bee, and we have the drones. So the unfertilized eggs, they become the drones. Whereas the fertilized eggs, you know, they will become the queen and the worker bees. So such type we can appreciate in the honey bees, this haploid diploid method, even in ants also. Able to follow, ma? Okay. So now we will go to the sex determination in case of in case of humans. So when you take the sex determination in humans, we are having the XY type of determination. So here we are having usually 23 pairs of chromosome. In that 22 pairs are autosomes, and that will be same in both men and women. But they are called the autosomes. They are called the autosomes. And then if it is going to be a male, it will be XY. And that is, there will be two type of gametes that will be produced during spermatogenesis. That is 50% of the gamete, the sperms will be carrying X chromosome, and 50% of the sperms will carry the Y chromosome. Okay. Now in case of female, it is the same. We have that autosomes, and then we are having the XX. So it will have all the 50% to be X, another 50% of X. It's going to be the similar. Now, what will happen in case of fertilization? So, what will happen, say X? So, what will happen? AA plus XX, AA plus XX, AA plus XY, AA plus XY. Okay. So, it is, you have to understand here, with this is a male, this is a female. So, what is happening here? What is happening here? We are finding four the four zygotes that can be probably formed. So you can see that one X is contributed from the father, another X is contributed from the mother. The child develops into a female. So it is the Y chromosome which determines the sex of the child. You can see here the genetic makeup of the sperm that determines the sex of the child. So there is always 50% probability of becoming a male child or a female child. But still we know that who keeps on blaming that this is a girl child or a boy child? It is again the men. But who is actually responsible behind that? It is the men only. So you can see the Y chromosome is the one which determines the sex of the child. So we have to know that 50% of the sperms that carry will have X chromosome, and 50% that which carries the Y chromosome, they are called androsomes. Okay. Now you can see that the sex determination in case of human beings, I would like to explain. There is something called a gene, what we call it as SRY. So the sex determining region which is located in the Y chromosome. This particular factor is absent in female. So they say that this SRY gene is mainly responsible for the testis formation. So that you need to know. So there is sex determining region in the Y chromosome. That is the one which is responsible for the testis formation, and again that factor is getting absent, then the embryo will all probable will develop into a female. Okay. Now we will just look into this question. Here the question is asked how is that the sex determination mechanism different in birds? Yes, they are different in birds. Now we will see the parents. Okay. So here we are having the female, we are having the male. So we will take the chromosomes. Okay. So what could be here? They are AA plus ZW. We are talking in terms of birds. First in birds, what it is? ZW, ZZ method, right? So here there are two autosomes and the male type. So what are the chances of those gametes? They can be A or A plus Z, or it can be A plus W. Here also A plus Z, and it can be A plus Z in case of males. Now how will be their offsprings or the progeny? So the chances will be what? AZ, okay, plus AZ plus this. Okay, so what will be there? So if it is going to be A plus Z W, okay, AA plus Z. So what you have to understand from here? So if it is going to be both similar homogamete, then we call, if it is going to be similar, we call this to be developed into a male. Okay. And here what is going to happen in case of, if it is going to be in the case of Z and W, it is going to develop into a female. So you can see the sex determination mechanism is quite different because females are heterogametes. Females are heterogametes, and you should know which is going to be responsible for the sex of the chick. It is the egg, because egg is only having the heterogamete, and that is having all the possibility to determine the sex of the chick. Able to follow, Aditi? Any doubts so far? Shall we pass to the next interesting session, the mutation? Now coming to mutation, it is nothing but it is an alteration in the DNA sequence. So what is happening? We have the entire sequence of the DNA. So somewhere there is getting a change. When there is a change in the particular genotype, definitely there is going to be a change in the phenotype of the organism. So in addition to that, we have those recombination. Mutation is also one such that is giving variations in the DNA. So what are all possible that we can have? So the what are all possible like we can have, we can see the mutation that we can see in the form of chromosomal aberrations where we can observe in the cancer cell. So there can be a particular what you call a sequence, it can be lost, or they can be duplicated, or they can be inserted somewhere, or they can be reversed. So then what happens? There is definitely alteration in chromosome, and that what alteration in chromosomes we call it as chromosomal aberrations. And this aberrations, quite example I can give, it is seen in the cancer. Okay, so the characteristic of mutation, like we can ask a question whether it is good or bad. So when it is put on to the pathogenic side, like I'll say it's bad, but on the other contrary, like it provides a raw material for evolution. So it is the ultimate source for all the genetic variation what we observe also, and that is where we need to understand regarding this. So the chemicals that which bring about these mutation, we call them as what is called the mutagens. So the mutagens can be chemical, it can be physical, even it can be biological. So we can keep on saying regarding those mutagen. So the mutagens here, they induce the mutation. Like we have UV radiation, gamma radiation, they are all quite good examples of mutagen. Now you see that there is going to be a sequence here. If there is going to be a sequence, say A, T, G, so this is the usual sequence. Suppose if there is going to be a issue with only at one point, okay, there is a mutation arising in that particular one pair of that particular chromosome, what we see, okay. So then we call that as what is called the, we call them as the point mutation. That is called the point mutation. Okay. If suppose there is going to be this entire part is getting completely reversed and put up on the other side, or they are getting deleted, or something new, a frame is getting inserted in between. So we call such things as frame shift mutations. So most commonly we have either a point mutation or we can have a frame shift mutation. Okay, so that you need to know. So when it comes to the mutations, actually we say there is something called germline mutations and somatic mutations. When I say germline mutation, the mutation that will occur in the gametes, that is the germline cells, that is going to lead to the formation of the gametes. Okay, but here when I say somatic, here they are totally occurring in the somatic cells, okay, which are non-reproductive. So like that we have the mutations. So when we speak with the mutations, like when I say the genetic mutation, I said there are many things, like based on the structure, like it can be a point mutation where a small particular structure of that part, at one point there will be a single base pair change of the DNA. Okay, like example we can give sickle cell anemia, or it can be a frame shift mutation like deletion or insertion, as how I've spoken to you all about. Okay, if there is going to be change in the number of chromosome, if there is going to be change in the number of chromosome, we call that as heteroploidy. We call that as heteroploidy, and there are many things beyond this. We have what is called polyploidy and aneuploidy, which I will keep explaining to you as it comes here. Okay. Now we'll go for next interesting thing, Okay, what we call it as, what do you call, I'm going to explain you another interesting subject, that is the pedigree analysis. I think I'll just explain here itself you with the thing. Okay, just 1 minute, I'll explain. See, one thing it can happen, either a chromosomal number can change that results in ploidy, or there can be change in the structure of that particular gene, like a mutation which is arising a change in that particular base pair or the entire sequence of the base pair. So we saw the two type of mutation. And 1 minute, if it is going to be say for example gene mutation, it can be either frame shift or a point. And if it is going to be with a number, I said that heteroploidy. What is heteroploidy? Heteroploidy is the change in the number of chromosome. Change in the number of chromosomes. So it can be either a polyploid, okay, or it can be an aneuploidy. So when I say polyploidy, here it will have more than 2 sets of chromosomes. Okay, so they will be like triploid, tetraploid, like that. If I say aneuploidy, then it can be either hyperploidy. So aneuploidy is actually a numerical change you see in a single chromosome. So it can be hyperploid or it can be hypoploidy. So aneuploidy is nothing but a numerical change in a part of a genome, usually a change you see in a single chromosome. If I say hyperploidy, here the chromosome number, that particular number in that organism will be higher. Say like trisomy, Down syndrome, trisomy 21 is a hyperploidy case. If I say hypoploidy, the number of chromosome in that particular organism is lower. Say like monosomy and all, where it will be less. Okay, able to follow, Aditi? So this is a quite little bit description about what you understand by polyploidy, aneuploidy and all that. Now we'll go to the basic of those what you call the pedigree analysis, as we see. So what are pedigree analysis? Actually the inheritance of traits we can study using the pedigree charts, like how it is getting inherited and how a particular trait is being passed from in a family, it's like a family tree, and how it is passing from one generation to the other generation, particularly by making use in the case of genetic disorders. So the disorders are inherited, which you all know. So the analysis of that inheritance pattern, that we can make use using it. So such an analysis of traits in several generations of family, we can use it what we call the pedigree analysis. Like it's analysis of a particular trait you are going to represent in a family tree over generations. Like you can see here how it has been made. So you can see, like you can actually trace a particular inheritance of one specific trait or some abnormality or some genetic disorders. For that you can use. So here you see here, the square ones are usually the males, the circle ones are the females. If we are going to put this rhomboid like shape, it's sex unspecified. If you are going to color it, the color is not specified, so that if they are going to darken it, it is the affected individuals. The single line shows that they are being related, got married, like that. If they show double lines, it shows the consanguineous marriage, like mating between, like getting married within the cousins, like that. So here if it represents there, like it's a male and a female, they get married, the father, mother, okay, and then this shows the order of birth. Always we should read from left to right, and you can see that if I show like this, it shows the parents with affected. This means the 5 unaffected offspring. Okay, these are some of the common symbols that we use. Now before going into this, I would also like to tell you like how to read a pedigree chart. Like before going into a pedigree analysis, first and foremost possible things that you need to remember is, we are going to have the chromosomes that I said that we are having 23 pairs of chromosome. In that we have autosomes and we have what are the allosomes or the sex chromosomes. So the probable thing is 1. So we can make the disorders as say it can be a dominant or it can be recessive, and at the same time it can happen in an autosomal chromosome or it can happen in an X-linked chromosome. So when I say, so it can be in these four combination, like it can be an autosomal dominant, or it can be autosomal recessive, or it can be X-linked dominant or X-linked recessive. So these are the four combination of things that we are going to look through. So in the exam, they will ask you about these things, where I am going to give you few tips that you need to follow. Whenever you are looking into a pedigree chart, okay, these steps should be made. So always you should look from top to bottom. Whatever I am mentioning in the first, I am showing about the parental generation, the first generation. The second line always depicts their children, and third line tells about their grandchildren, and so forth. So it is the first, second and the third generation. So I said probably we can study this in terms of autosomal dominant, autosomal recessive, and at the same time X-linked dominant and X-linked recessive. Now we will see about the autosomal recessive. What is a classical example that we can give with autosomal recessive? The best example we can give is the sickle cell anemia or the thalassemia, which we will see quite faster in the next coming sessions. Now you have to understand here, I said this one, what you see here, okay, this represents a male and this represents a female. These are the offsprings and all that. If it is shaded, that means it's affected. So here 1 and 2, you can see that's a father and a mother, and this shows the children that is born to them. Then this is one family. Okay, so 1 and 2, they are having 6, 7, 8 as their children. 3 and 4, this is another family. Like that you need to think. So 9, 10, 11 are their kids. 9, 10 and 12 are their kids. Okay, so that is how you need to understand. First, when you are looking into a chart, first look into how many sexes and how are the frequency in which they are getting affected. That will give you the first nice point. Here when it is going to be cases like autosomal, we can see that both the sexes will be equally affected. But when it comes to a X-linked type of disorder, there the proportion of males affecting will be more than the female, because female will have, even though she might be a carrier, she will have one X normal and she'll have a defective X. But here in case of male, they'll have only one X, and that X also is defective. Okay, so that is why more males will be affected. So we will see first about the sexes. Like you see arbitrarily, like 1, 2, 3, 4, here 1, 2, 3, 4, like more 1, 2, 3, 4, 5. Like it's more or near to equal frequency. So it appears in both the sexes with equal frequency. And when you observe here, you see here in this, they have shown the one thing, what you understand by autosomal recessive. That means it occurs in the autosomes, that is the somatic cells. When I say the word recessive, first you understand, I'll give one example. Say for example, if I'm using the word A, okay, and there are like this A. Now, when I use the word autosomal recessive disorder means, then children with this, both the A's and this, they will be normal. Only when they have this homozygous recessive, then they will be having the particular disease. Okay. So you can see here in this case, what is happening? Whenever this recessive is being expressed, they are going to have the disease. Now you look into this family 1 and 2. Okay, Aditi, they are the parents. What happens here? The trait is going to skip the generation. You can see that it skips the generation and only it appears in the next generation mostly. So always when you look through the affected individuals, the affected offsprings, they are mostly born to the parents who are unaffected. If the parents are heterozygous, say for example, if they are, say for example, here, if we are showing this colored one, okay, so this is shown, so it can be either AA or it can be AA, which will be quite normal. Only when it is AA, they will have the probability of developing the disease, right? So here, that means what is happening here in this case, it would have been AA and here it would have been AA. So that is why it skips the generation, because it is recessive. But what is happening here, as in this case, when they are getting married, here the parent would have been either AA and here the one of the chances will be having AA. So there is all the chances to have the expression of this homozygous recessive, and that is why the what you call That is why there is a chance for the progeny to develop the disease. Okay, Aditi. Now, Aditi, I am audible. Yes. So now what you can see here is, so mostly they say that the affected offspring, they will be having parents who are unaffected. But if the progeny is born, okay, that means if the parents are heterozygous, approximately they say that 1/4 of the progeny, that is 1/4 of the progeny. Okay, so here you imagine, okay, so here what are all the probability? It can be like this, it will be like this. So that is why they skip the generation. But what will happen if he gets married to another? Then there is all chances of them to develop this particular thing. You're able to follow. So that is where. So what is happening? The heterozygote individual, approximately 1/4 of the child, they can have this probability, like you can see here. So you can see that approximately the heterozygote, 1/4 of the progeny might be affected. And this will be very quite common with the consanguineous marriage. Like you see here, you can see that one of the parent might be a carrier or they might be heterozygote. So what will happen? Definitely, as they see, okay, mostly they will not be affected. If at all affect, they might be having a heterozygote parent. Ma'am, is there a probability even when the parents are affected, would their offspring be unaffected? Yeah, that is there. So now Aditi, do not audio and also everything is clear. Fine, thank you dear. Now Aditi, you forget about that first question what you have asked. Wait, I am explaining you with the condition of autosomal recessive. Autosomal recessive means what? This should be in a homozygous recessive trait, then only the disease is going to get expressed. If it is going to be in the heterozygous condition, okay, that is if it is going to be homozygous dominant or in this heterozygous condition, what is happening? They are not getting expressed. They might be a silent, they might be having that recessive. That is why it skips so many generations. But if they are going to have consanguineous marriages, they have all the chances of getting exposed to the same set of that particular gene. That is why in one of the generation that will get expressed and they are going to have the disease like sickle cell anemia or thalassemia. Okay. So mostly when parents are affected, that we will come in dominant. See, you're asking, ma'am, is there a possibility even when the parents are affected, would their offspring be unaffected? Yes. If the parents are affected, here sometimes they might be having that heterozygous trait. They may not pass. They will also be in heterozygous state. Okay. So only when it comes to another generation where there is a marriage of the first cousin or there is a consanguineous marriage, then there is all the probable chances for them to have it, and either the third or the fourth generation might be having the possibility. Okay. Sometimes it may not skip the generation, because if it is a heterozygous parent, then there is all the chances of that to get expressed 1/4 of the progeny. Okay, they might have the chances to have that. If you see all they will be in heterozygous, then where all is that they will be totally expressed. Like when you see both the affected parents when they get married, see here they are having AA. When they get married, then there is all the chances of them to be there. That is why, see, you understood. See here it will be like this. Okay, so there will be all chances like to have is in this case. Okay, and if the other person from outside who is getting married also, if she is like this case, like heterozygote, then what is happening? Definitely they are going to have the recessive trait, and that is why the child is going to suffer from it. Able to follow, Aditi? Able to follow? See, in this family we are having the parent. Okay, so they are heterozygote for it. Okay. Now what happens? All these kids, they don't get affected because that skipped the generation. Now if he is going to marry to another person who is also same, they might be affected or they are heterozygous, then definitely one of the child will be the sufferer of the disease. Okay. If that is what we are telling, they might be heterozygous, or else they will have it, but it will skip the generation. As I said, it will skip the generation. But if he is going to have a consanguineous marriage or something, then probably you can see. But here you see how nicely it is coming up. So once when this comes, they will be affected. So here also the same thing. Okay. So what is happening here? These two are getting married. So what is all the chances here to have it here? So they will be like AA. Okay. So here also it will be AA, and this will be a heterozygote condition. So here, So it is not in this two families, these two people are not exposing. You look into that 8 and 9. Okay, they are from family who had this, but they are not having. But what is happening? They are, you can see that the offspring is affected. The 17 number is affected. 8 and 9 are not affected. Okay, so the affected offspring is always born to the unaffected parents. So you can see that they are, so here also you will have the chances like this only, AA. Okay, so that is where the thing. So what will happen? Definitely one of the child will be affected. Able to follow, Aditi? Yes? You are able to follow? So imagine this will be like this. So they will be mostly like this. So what will happen? So when you make across a thing, so now this 2A, when it comes only, the child is affected. You are able to follow. Okay, so they skip the generation. 8 and 9 they didn't have. Okay, but what is happening in the other set of generation? This is getting affected. That is where you need to understand. Able to follow? Yes, Aditi? Okay, so this is where you need to understand. If we are going to see about the autosomal dominant, you should see here, first I always said, please take up the look of the sexes. Here you see one family, 1 and 2, 3 and 4. First you make the generations. Second, always from left to right, look for the sexes. Yes, they are equally affected. That means it's autosomal. Now you can see that here both the sexes, they transmit their trait to their offspring. That is another important thing. Okay. So as you see here, the traits, they are going to pass out it on to their respective offsprings. So here it is not skipping the generation, and every affected child is having an affected parent, either one of the parent. Okay, when one parent is affected, if I say autosomal dominant means, say the same gene if I am taking. Autosomal dominant, we can give example as Marfan syndrome. Good example can give Marfan syndrome or Fragile X syndrome and all we can give. So look here, if I say for a particular gene, that means now only with this AA they will be normal. If they are having this or this, that means what? They do suffer from that particular autosomal dominant disorder. Now when you look for the particular thing, here see both the parents, they are affected. Okay, so what happened? They are just going to pass the trait to it. So here the children are affected. That means they will carry that dominant genotype. That is, they will have the dominant trait here. Here, that means what? Here the child will be having the the recessive. That is why it is not expressing here. Okay, so you can see here that there is always a chance for a heterozygous parent where he is 75%, then he has all the 25% to give it to their children. Okay, if it is going to be dominant, say AA, then 50% chance is there exactly to pass on to their offsprings. Here it is not skipping generation. Look into 1 and 2. So definitely the 5, 6, 7 are their kids. In this, see, the affected offsprings, they have their affected parents. Now when only one parent is affected, the other is unaffected, then what happens? Approximately half the offspring will be affected. So it is 50-50%. That means what? If it is going to be of this trait, okay, so here the one of the parent is what? It's unaffected. Okay, so definitely what is going to happen here? As you all see here, you can see only one is getting affected. So it can be, okay, the other is unaffected. Okay, so unaffected parents usually do not transmit the trait. Only the affected parents, they pass on. Now look into this condition. Okay, now the affected parents, that is the unaffected parents, they don't pass the disease to the offsprings. Only the affected parents, they pass on the trait. You are all able to follow, Aditi? Yes. Now coming to the X-linked dominant. Okay, X-linked dominant. In X-linked dominant, as I say, here only we have to say about that Rett syndrome and all. In this, I said it is X-linked. Please recall that X-linked. Here, when I say X-linked dominant, okay, here you can see that often here the more the females, mostly both equally they will be affected. Both males and females will be affected. But often you can see more females will be affected when compared with the males. So that is where, because it's X dominant. Who is X dominant? It is a female. Here you can see that it is 1 and 2, the family, they get married. You can see that it is not skipping generation. Now, first thing when you look into a pedigree, I said first look for the sexes, whether they are equally affected. Second point, you are going to look for, what is the second one, did I tell you? First, make out whether it is autosomal case or X-linked. Second thing, when you are coming, you have to look for the whether it is skipping the generation. That gives a very important clue for us. So here see, it does not skip the generation. And all the affected sons, you see, they will have affected mothers. Okay. And all, so here they have a son, a daughter, they have a son and a daughter. So you can see that all the affected daughters must have either an affected mother or an affected father. Here you see, here all these are the daughters, these are the sons. So definitely she will have either an affected mother or an affected father. And always you should see that the affected fathers will pass the trait on to all their daughters. So always it is the father, because she has to have this both X. One X she is going to get from the mother. The other X she has to take it from the father. So the one X which the father has itself is defective. Then what is going to happen? It will just come to the daughter. So all the affected fathers will pass the trait to their daughters. And affected mothers, when you see, if heterozygous, like how we saw, she will pass half to their sons, half to their daughters. So if they are heterozygous, because it is X-linked dominant. Okay. So she will have 50-50 of it. So she will pass only the she will pass only the 50% of it, and the other 50 to the daughters. So this is where you need to understand about the X-linked dominant. Now X-linked dominant, like we can say conditions like vitamin D resistant rickets, or it can be a case where that Rett syndrome, all that we can take examples. So it is always a male who will pass the trait to their daughters and none of his sons. That is one big clue. So the father will pass to the daughters and not to his sons. Another thing, a female with this particular trait, she will pass on to all the sons and daughters. So every affected person will have at least one of the trait from the parent. If the trait disappears from that particular pedigree, okay, it does not reappear over a large number of pedigrees, and there will be more females when compared with the males. So that is where we need to understand. So how you make it as X-linked dominant? First look in whether how many males or females affected. More female, X-linked dominant. Then look for it is not skipping generation. Always if a mother is heterozygous affected, she will give it to both daughters and son. If father is affected, he will pass it only to the daughters and never to the sons. This point you should know. This will give you a clue about X-linked dominant. When I say X-linked recessive, this is quite good. We know the condition like hemophilia and all that, right? So they come under this particular condition. Here you can see that more males will be affected than the females. So you see the family 1, the female 1 and 2. So they are getting married. Okay. So here they have all the, suppose for example, here the female. Okay. So here she is a carrier. Okay. She is carrier. So she might be having, say, X. This is a normal male. Okay. And here this is a female whom she is, say she is heterozygous. She is normal. She is normal. Okay. She is a carrier. Now what is happening here? More males are affected, and always the affected sons, they are usually born to the unaffected mothers, because the mothers will be silent carriers. And you can see the trait will skip the generations. And approximately you can see that half of the carrier mothers, the sons will be affected, and it is never passed from the father to the son. So what you are seeing, she is an heterozygote, that is heterozygous normal, that means she is a carrier. So what are all the chances they will be here? So here she will give this one of this X, okay, and this X will be taken from the father. So definitely what will happen if it is going to be like this, say it is going to take the Y from the father. So say this X from the mother and this Y from the father, and that is why it is hemophilic. Whereas the daughters, you see, they will take this X from the mother, suppose example, and she might take one from here, or she can take one like this with one. Okay, so there is all these chances. So that is why you can see the sons who are affected, the mothers will be the carriers. So it never goes from fathers to sons. Okay, and you should also know that another thing, and all the daughters who are affected, you see the daughter here, she is there. Now you see this is a family, they are having kids like 4, 5, 6, 7. Here the sons are affected. Okay, now when you see the sons are affected, here the son is affected, so he is giving birth to the daughter. The daughter will be affected because the father is a carrier, because she will get it from the father. So that you need to be knowing. So all daughters will get from their, all the daughters who are carriers will get from their father. Okay, and if the son is going to be a sufferer, it has to be carried from the mother. So how is that we are going to determine here? First, always look whether it is autosomal dominant or what you call X-linked. So quietly look into whether it is male or female that is affected. Then look for X-linked. Then look for the crisscross inheritance, whether sons from sons to daughters or mothers to son, father to daughter, mother to son. That crisscross pattern you look for. Then if it is 50-50, then it is autosomal. Then look for whether it is dominant or recessive. So for dominant, what all the tips I told you, if it is going to be dominant, then you should see the dominant, it will not skip the generation. Okay, it will not skip the generation. It does not skip the generation. But if it is going to be trait, if it is going to be recessive, then it is going to skip the generation. That point you have to be knowing. Okay, and then skipping the generation will take place only in the recessive. So check the ratio. So look for this. So if the disorder is dominant, one of the parent must have the disorder. If it is going to be recessive, that neither the parents have the disorder because they will be heterozygous. So there will be skipping of generation you can observe in recessive. So check the ratio of the affected individuals. If skewed to one type, then X-linked, 50-50, then autosomal. Check for that crisscross. So skipping of generation means recessive. If not, then dominant. Like that you have to go stepwise to find out. Okay. So very quick, first look whether it's skipping of generation. By looking into skipping of generation, you will know whether it is going to be or whether it is dominant or recessive. Second, looking for crisscross pattern, you can find out whether it is X-linked or autosomal. With that, you can narrow down your steps also. Now here you look into it. It is evident that this is a X-linked recessive trait, that the transmission is from a carrier female to a male progeny. So all these that you know, like you can see the pedigree, they are showing for the autosomal dominant like myotonic dystrophy. So what is that with the autosomal dominant? So see here it is equal in both the sexes, 0.1. Here you can see that it does not skip the generation, right? Here you can see that both sexes are affected, appears in equal frequency, and always see that affected offspring must have an affected parent. Okay, and another thing, you have to know that thing, when one parent is affected, okay, generally, then the other parent is unaffected, then half of the offspring they will be affected like this. Okay, 50-50. And unaffected parents will not transmit the trait. So this will give you the idea about the autosomal dominant. So autosomal dominant, it is not skipping generation, 0.1. Second thing, you have to know autosomal because there is no crisscross inheritance. So you can see that either of the, if the offspring is affected, it is coming from the parent, and it is not passing from unaffected parents will not transmit the trait. That is a very good significance of autosomal dominant. When it comes to autosomal recessive, trait like sickle cell anemia, you can always say it also appears in equal frequency, but it will tend to skip the generation. Okay, so you can see that here, see, it tends to skip the generation. But what is happening here? The affected offspring always, okay, so you see, the affected offspring will have unaffected parents. Okay, so that point you have to know. Okay, and if at all, if the parents they are heterozygous, then 1/4 of the progeny they can be affected. That point you should know. And it's more common with the consanguineous marriage. Ma'am, then X-linked dominant is autosomal dominant. No. Autosomal dominant is quite different from X-linked dominant. Autosomal dominant means, please understand that autosomal dominant, I'm talking with the autosomes. Okay. So their autosome dominant means like we get it in terms of, say, here. Okay. In autosomal dominant, like suppose if it is present in these conditions only, then the disease is going to be expressed. If it is in this particular, then they are normal. Okay. X-linked dominant is different. It is appearing in the gamete. It is transferred through the XX chromosome. So it is with XX. Okay. X-linked is different. X-linked is different. You're able to follow. X-linked dominant is quite different. Okay. I'll just go back. See here. So in X-linked, what I said here, that means what I'm talking about, XX and XY. So it is X-linked dominant. So if the female is going to have both, then they are going to suffer from that particular disease. So that is why here the more females will be affected than men. That is where. Okay, able to follow. So then we have what is with, now I have given a practice question. See which one of the following symbol and its representation is used in human pedigree is correct. Just look into it, which is correct. Please put the option, Aditi. Yes, you are right. Yes, you are right. So which of the following is correct? Because this we don't use. We use this, right? This is an unaffected female. We use affected male with this color. Unaffected will be like this. Unaffected female will be just round. Unaffected male will be like this. So this is the correct one, mating between the relatives. So that is the right option. Which of the trait? Autosomal recessive, sex-linked, Olendric, which is shown here. So the heterozygous individuals have not been indicated. So just show this. Yes, very good, Aditi. What you have answered is right. What trait is this, ma? As you are seeing here, which type of trait is this? So first you should look for what? We have to look for the number of individuals they are affected. Second, look for whether there is skipping of generation. Then you look for the people, like how they are getting affected or not. Just look for it. Yes, you are right. It is autosomal dominant. So how you will say that it is autosomal dominant? Now see that it appears in both sexes of equal frequency, 0.1. It does not skip the generation. Acha. Now affected offspring will always have an affected parent. And if there is going to be say like an heterozygous parent, okay, then what if it's heterozygous, then we can say that 50% is affected, and it will never come from unaffected parents. You see that all the affected offspring, they have an affected parent, affected parents having affected offspring. So that will give you an idea about autosomal dominant. Okay, this is how you should check. Now we all know that the DNA, okay, that is any organism, okay, they are controlled by gene. And where is the gene located? In the DNA. So the DNA is the one which is going to give the information regarding the genetic information. If there is going to be any alteration in the genetic material, superb, Aditi. If there is going to be any alteration in the genetic material, we call that as mutation. So there are a lot of factors that they have been associated with inheritance or altered genes or chromosomes that they have associated. We'll see what. So genetic disorders we can classify as chromosomal disorders or Mendelian traits. 1 minute. So like we have the chromosomal disorders and we have what is called the Mendelian traits. Now when I say the Mendelian traits, we have autosomal disorders, like I said, the autosomal recessive like sickle cell anemia, thalassemia, or it can be sex-linked disorders like hemophilia, color blindness, okay, and like in aneuploidy or polyploidy where we see Turner syndrome, Klinefelter syndrome and Down syndrome. So now when I say genetic disorders, you can classify as Mendelian disorders and chromosomal disorders. If I say Mendelian disorders, there is going to be an alteration or mutation in a single gene, okay. So they are going to be transmitted to the offspring, same like the lines of inheritance, what we studied in principles of inheritance. So you can easily trace out by using a pedigree analysis. So the most prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria and thalassemia. But you have to know whether they are dominant or recessive. Depending on that, we have to know. Whereas in this, the Mendelian disorders, this hemophilia, it is a sex-linked recessive disorder. Here what is happening in hemophilia, like you can see that hemophilia, it's a bleeder disease. It is actually happening where in a person, if they are going to get a cut or an injury, the bleeding will not stop. Okay, there are two types of hemophilia. We have hemophilia A and hemophilia B. Hemophilia A is due to the deficiency of factor 8 and hemophilia B is due to the deficiency of factor 9, clotting factor 8 and clotting factor 9. That is why it causes hemophilia A and hemophilia B. Now you have to know that in usual genotype, okay, which I will explain. So here we have to know that mostly the females will be carrier and they will be passing on to their sons who will become a sufferer. So the females will be carrier and males will be the sufferer because it is X-linked recessive. Mainly it is X-linked recessive disease, X-linked recessive. So they say that in Queen Victoria family they were having regarding this. So in this particular thing, the clotting of the blood is the one which is getting affected. I will explain you this, please listen to it. So the genotype I am writing, suppose in males you see, they will have this, if they have, they will be, usually they are normal. If they are hemophilic, it means it is recessive is present, then they will be suffering from hemophilia. Okay. If it is a female, okay, she will be homozygous. That means she is normal. Okay. Sometimes she might be heterozygous, where she will be a carrier. Okay. She will be a carrier. This is hetero. This will be homozygous normal. Homozygous normal. This will be heterozygous carrier. Okay. Mostly females will not be affected and it is a very rare condition. Now I will explain you, since you can see the entire of the thing is getting inherited in the X, that is why we say it is X chromosome is the one which is affected. Now I will explain you the crisscross in inheritance. Now you see, now the father is hemophilic. Okay. We have a mother, she is normal. Okay, now they are getting married. Okay, now as they get married, what are the probable things we can get it on? So they will have XHX, XHX, XY, XY. So here in this children, you can see as with X-linked disorder, it's here the females, they will be the carriers, but you can see that it skips the generation. Second thing, it's recessive, that's why it skips the generation. And you can see that here the females. So naturally you can see here that it always passes from the father to the daughters and never from the mothers to the daughters. So it comes from father to daughters and from mother to sons. So that crisscross is there. And next, what happens to their grandchildren we will see. So in their grandchildren, what is happening? Suppose this fellow is getting married to this woman, she is getting married to another normal man. What all the possible they can have? They can have XXH, XHY, XX and XY. So she will have a carrier daughter, she will have a hemophilic son, and this will be normal and this will be normal. So you can see that it skips the generation. So here there is one child with hemophilia. So it skips the generation. Okay, here the grandfather had, the grandson is having. The father is not the sufferer here. It skips the generation, recessive. Second thing, you can see that it is always inhibited in the crisscross pattern. So it is a crisscross inheritance we say. It is a crisscross inheritance. That is, it is always, the character is inherited to the second generation as the first generation will be the carrier. So this crisscross inheritance, and always it passes from the father to daughters and mothers to the sons. Okay. So that where you should know. So can you see that if father is hemophilic and mother is homozygous normal, predict the genotype of children and grandchildren. So how is that? So when you are going to make it, this will be there. See here, the father is hemophilic, the mother is normal. So you can see here the children, that is the daughters, they are all carriers. Okay. And what about the sons? They are all normal. But what happens in the next generation? If they are going to get married to a normal female, here you can see a daughter is a carrier, the son is the sufferer, and they have two children who are normal. So this is how it happens in the crisscross inheritance, which is characteristic of X-linked recessive disorder, particularly the hemophilia. Able to follow, Aditi? Now coming to the colorblind persons cannot distinguish red and green colors. Probably this is very important in case where those people are getting appointed themselves in signaling departments, telecommunication departments like railway, pilot service, traffic police and all. So it is also most common sex-linked disorder. This is also expressed in recessive condition. I will explain you, ma. So always you should know that the homozygous recessive in female, the homozygous recessive female, okay, they will be like this XC, XC. whereas that is the heterozygous recessive male will be X, C and Y. Okay, so the inheritance, it can be by two ways. One, you see the parent. Now here imagine the man is a colorblind. Okay, so I'm going to show you the pattern between the colorblind men and a normal women. Okay, so what will be the parent? Colorblind man. So colorblind men will have XC and a Y. The women will have both X normal. Now what will be their generation, first generation? They will have X, XC, and they will have X and Y. So here, even though she is normal, she will be a carrier of the disease. Then what happens in this? Now this XC is getting married, okay, to another normal man. What will happen? Okay, the second generation, you can see that here there will be normal daughter, okay, a son who will be normal. They will have XC, but here the son will be a colorblind. Okay, the son will be a colorblind. Okay, so always you can see that here this is a male and this is a female, right? So here the female, she is getting married to the normal, the female who is a carrier, she is getting married to a normal male. So what is happening? You can see that the children here. So there is a normal female, here there is a normal male, a daughter will be a carrier, but a son will become a colorblind. So you can always see it comes from the mother to the son, okay, and also from the father to the daughters, okay. So that crisscross pattern of inheritance you can observe even here in this pattern, okay, because it's an X-linked recessive disorder and it always skips the generation also. Now I will explain you here another thing, like you can see here, if it is going to be between a okay, if it is going to be between say a normal vision men and she is going to get married to a color vision blind women, okay, what is going to happen? So the first generation, they will have a female who will be a carrier and they will have a son who is a color blind. Okay, if the mother is color blind, okay, definitely the son will be color blind. Okay, here the father is normal. So this is the first generation I'm speaking. Now this daughter, if she gets married to a person who is colorblind, okay, who is also a person who is what do you call a colorblind person, what will happen? So here you can see XCX, okay, and then XC, then they'll have XX, and then so you can see here one child will be normal, but she'll be a carrier, a female child. So you can make the crisscross inheritance, this pattern I'm showing. Okay, so like that. So XC, XC, so you're getting it. So here a daughter will be colorblind and then a son will be a colorblind. So the daughter will be colorblind, the son will also be colorblind. The son will also be colorblind, as you all see. But one carrier and only one normal male child. So this is how the pattern of inheritance will be there if a colorblind mother marries a normal man. So if the mother is colorblind, she will definitely give it to the son. Okay. And then what will happen if they get married, and the daughter will be a definite carrier. So if they are going to get married also, what will happen? Definitely there will be carrier and a daughter will be a colorblind. So that so if the grandmother is colorblind, definitely the grandchildren, both granddaughter and grandson, both will be colorblind in a probable ratio. That you need to know. So you see here a normal woman whose father was colorblind is married to a normal son. The sons would be 75% colorblind, 50%. So you're taking a normal woman, she is marrying to a colorblind man. So what will be the probability of sons to be colorblind? Yes, you're right. It will be 50%. As I said, because 50%, because the father is a colorblind. So he always gives to the father will always give it to the daughters. Okay, and the son, from the mothers it goes to the sons. So you can see that there is always a 50% chance for to be the sons would be colorblind. Okay, so whose father was colorblind is married to a normal man. Okay, you understood? There's a normal woman, but whose father, that means the grandfather we are talking about, he was a colorblind. Okay, that means this woman would be a definite carrier. So when she is married, what the sons they will have, that means the grandchildren, 50% will be colorblind. Okay, if the female child, they will be carriers. Okay, so that point you should know. Okay, so this XCC, which is she's a carrier getting married to another normal man, 50% chances of getting colorblind. So coming to sickle cell anemia, which I already explained, it's an autosomal linked recessive trait. It comes from parents to offspring. Okay, so here it is an autosome linked recessive. I always said, if I say recessive, that means only the recessive thing when it is present. Then if it is going to be in heterozygous, when both the partners are carrier for the gene, if it is in the heterozygous state. So the disease will be controlled by a pair of allele, HBA and HBS. Okay, so if it is homozygous, they will be in this form. That will show the disease phenotype. Okay, so heterozygous individual, they will be mostly unaffected and they will be carrier because 50% probability of transmission. What we call it as sickle cell trait. So usually in the beta chain, in the 6th position, the glutamic acid will be replaced by valine. So whenever such a thing happens, there is single base substitution. See, from GAG to GUG, that small point mutation is the one which is taking place. So when RBC, when they are exposed to such low oxygen tension, the RBCs will become polymerized and they will become like this sickle shape. So once when they pass through the capillaries, they get ruptured, leading to anemia. So see here, there is one small confusion. So in 6th position, instead of glutamate, what is happening? The valine is replaced. So they become sickle shape, leading to anemia. Then there is another characteristic condition called phenylketonuria. This is also autosomal recessive. It is an inborn error of metabolism. There is one particular enzyme which is not present, that is phenylalanine hydroxylase. So this enzyme, when it is not there, the amino acid phenylalanine will not be converted to tyrosine. So this phenylalanine will get accumulated and it will produce all the phenyl acid derivative, that is all those acids will get accumulated. So they will not be excreted in urine, and this accumulation in the brain will cause mental retardation, and the excretion through the kidney is also becoming very poor. So next, coming to the chromosomal disorders, I said already the change in the number of chromosomes, what we see in chromosomal disorders, it can be like because of absence or excess or abnormal arrangement. When I say aneuploidy, there is failure for segregation of chromatids during the cell cycle. So there will be either gain or loss. But like I said, the Down syndrome where there is extra gain of chromosome, or similarly Turner syndrome where there is a loss of X chromosome, like that. In polyploidy, there will be an additional set of chromosomes that is added up. The whole set will become extra. Say instead of diploid, they can become triploid, tetraploid, pentaploid. That condition we oftenly see in plants. So you see the chromosomal disorders. So normally we have 46 chromosomes, 23 pairs. In that 22 are autosome, one pair of sex chromosome. So you can see that there will be either additional copy or deletion of one copy, that like there will be loss of one copy of chromosome. So such situation, like we can say like trisomy or monosomy. One such conditions like we see with the Down's syndrome, Turner syndrome and Klinefelter syndrome. So in Down syndrome, you can see that particularly in chromosome position 21. So instead of 2 chromosome, you can see the trip copies. So what is happening? That's why it's called trisomy 21. It was first discovered by Langdon Down. You can see the baby, since there are 3 more the X chromosome, more the child will be mentally retarded. So they say that, see, the child will be mentally retarded. You can see the baby with a round head, furrowed tongue, broad face. The fontanelles will not be developed. They'll have congenital heart disease. You can see the big tongue, the palmar crease. They will have physical abnormality, mental retardation, psychomotor disturbances, etc. So that is with the Down syndrome. So trisomy 21. Please remember this. Then Klinefelter syndrome, here in this additional extra copy of chromosome. That is what instead of what is happening? In this case, the X chromosome is one additional. Instead of XY, there is one more chromosome in the sex karyotyping when they see. So what is happening here? Overall, they will be masculine, but in the case, there will be feminine development also, like development of breast. So there will be development of breast, and the individuals will be male because the presence of Y chromosome. So male reproductive organs, secondary sexual characters, all that will be poorly developed. They'll have gynecomastia, and some they'll be very tall, sterile, and you can see that it is trisomy of the sex chromosome 2n plus 1. So it will be 44 autosome plus XXY chromosome in Klinefelters. Okay, there will be sometimes mental retardation also. In case of Turner's syndrome, okay, in case of Turner's syndrome, able to follow, Aditi? Yeah. So in Turner's syndrome, like you see here, there is absence of one X chromosome. So it is 45 plus X naught. So it will be like that. So it will be 44A plus X naught. Okay. So here such females will be sterile. The ovary will be very rudimentary. Other features like it is like monosomy. Turner syndrome is monosomy, where it is 2n minus 1 of sex chromosomes. Okay, that point you need to understand. So there are totally only 45 chromosomes in Turner syndrome, whereas here there will be 47 chromosomes in case of Klinefelter syndrome. Here the female will be not having any secondary sexual characters. She'll be sterile here. So that point you need to know. So she'll have that webbed shoulders, short pinna, short stature, that is characteristic. Able to follow, Aditi? Yes, very good. Aditi, you have answered some questions. I didn't just see, just now I'm seeing. Very good. You're able to follow. Any doubts with today's class? Any other doubts? Yes. Aneuploidy is mainly because of non-disjunction of chromosomes during the meiosis 1. That is the main cause that you can remember. Yes. Shall I wind up the session? Shall I wind up? Able to follow? Yes. Okay. So thank you, students. Bye.